Good morning, students. This is what biopractical class 12, subject biology. So today we are going to discuss the practical or experiment effect of different temperature on the activity of salivary amylase. You know the enzyme salivary amylase and starch. Starch is a kind of polysaccharide, a kind of carbohydrate, a kind of polysaccharide. And tyrine or salivary amylase is a kind of what enzyme commonly called tyrine present in the saliva, and this acts on starch and breaks the starch into glucose. ठीक है, स्टार्च को ब्रेक करता है किसमें? ग्लूकोज में. So starch is a polymer of glucose. So on digestion starch is broken down into glucose. So this is the experiment. So effect of different temperature on the activity of salivary amylase or tyrine on starch. What the requirements in this practical? We require starch powder. We require starch powder. Okay. We need beakers. We need test tube holders, thermometers, ice cubes, tripod stand, split lamp, thermometers, and the chemical Lugol's iodine or iodine solution or Lugol's iodine. So iodine solution is used to, or Lugol's iodine is used to test the presence of starch. ठीक है? So what the test of starch? This is the first point. First line, starch solution. We take the starch solution, then heat the starch solution for 2 minutes, and after heating, add iodine solution. What happens? If the material is a starch, then it gives blue-black color after adding iodine solution. Okay? If it does not give blue-black color, it means it is not a starch. ठीक है? Now, starch can give the blue-black color on adding the iodine solution after boiling. If the starch is digested, तब क्या होगा? Suppose we have taken, in this case, enzyme has not been taken. Okay? Now, this is the second situation. We can put a star mark. In the second case, we have taken starch solution and then we have added salivary amylase or tyrine in the starch solution. Okay? And then what happens? The enzyme salivary amylase or tyrine, this acts on starch and it breaks the starch. What the temperature? We maintain the temperature at the rate of 37 degrees Celsius. Because tyrine is the enzyme which acts at our body temperature, and our body temperature is 32 degrees Celsius. Okay, so around 32 temperature is maintained. After putting the salivary amylase into the starch solution, what happens? Salivary amylase acts on starch and it breaks the starch into glucose. Okay, once the starch is broken into glucose, and then after heating, you want to add iodine solution into it will not give blue-black. Okay, in this case I have used the iodine solution. And it is not giving the blue-black color. What is the reason behind this? Because the starch has been broken into glucose, and glucose does not give. Glucose is a kind of monosaccharide. This does not give blue-black color on adding iodine solution. Okay? So this is the theory part of the practical. Next point is actual experiment is this one: effect of different temperature on the activity of salivary amylase. You know, enzymes are temperature specific. So we have taken 3 different temperatures. Temperature of ice. This is the temperature of ice. I have put very cold water into this, and there is thermometer also. Okay. Now we have taken 3 temperatures. Starch solution. First case, we have taken starch solution, and the enzyme tyrine is put into the medium. Enzyme is added in all the medium. All the 3 what contains enzyme in the starch solution. Okay, so this is the first case, starch solution plus what? we have put enzyme tallene into it, and this system is kept at temperature around 4 degrees Celsius, around 3-4 degrees Celsius, very low temperature. What happens at low temperature? At low temperature, enzymes become inactive. Enzymes are not destroyed, rather enzymes become inactive, and inactive enzyme, it is unable to break starch into, catalyze the breakdown of starch into glucose. Starch the glucose को break down नहीं करता है. Okay, so that's why starch is there. Starch is not broken into glucose, so starch is there. So when we put iodine solution in it, what happens? This gives blue black because of the presence of starch. Second case, okay, this one. In this system, we are maintaining the temperature 37 degrees Celsius, body temperature. So starch solution plus tallene enzyme in it, and the temperature is maintained. The water bath at the temperature 37 degrees Celsius. Okay, at this temperature, what happens? Enzyme becomes active. So the starch is broken into glucose, and the glucose, if we put the iodine solution, glucose is not giving the blue-black color. So glucose is not giving the blue-black color in this case. So it shows that enzyme is active and enzyme has broken the starch into glucose. There is no starch in the medium. So that's why it is not giving blue-black color. So activity of enzyme is taking place at this temperature, that is called 37 degrees Celsius of body temperature. Now the third case we have taken is starch solution plus enzyme tallene. Okay, in the water bath, third case, in the water bath, the temperature is around 72 degrees Celsius. 70 plus, maybe 72, 75, 76 or around 70 temperature we have maintained in the water bath. In this case, what happens? This is the high temperature. At this temperature, our body enzymes cannot function. So at this temperature called 72 degrees Celsius temperature, enzymes get denatured. Enzyme is denatured. Enzyme is destroyed. The catalytic side of the enzyme is destroyed, and that's why no breakdown of starch into glucose. So again here also starch is there. And when we give the iodine solution, we put the iodine solution into it gives blue-black. Okay, so in this case also we are getting the blue-black color, but after 20 minutes, after 15 minutes, 20 minutes, the color will gradually change. So enzyme is there. Enzyme has not been destroyed, but the enzyme action has been slowed down. Enzyme action slow हो गया, बहुत धीरे-धीरे काम कर रहा है. इसलिए हमें 2 मिनट के बाद, 4 मिनट के बाद में, 5 मिनट के बाद में ब्लू ब्लैक कलर दिखता है, लेकिन 15-20 मिनट के बाद में ब्लू ब्लैक कलर आपको थोड़ा कम मिलेगा. ठीक है, थोड़ा आयोडीन कलर आपको मिलना शुरू होगा. कलर चेंज यहाँ पर थोड़ा कम होगा. इस केस में कलर चेंज क्या हो जाएगा? आफ्टर 10 मिनट्स, आफ्टर 5, 6, 8 मिनट्स, 10 मिनट्स, ओके, you will not get the color because glucose starch is broken into glucose. They know after 10 minutes, after 15 minutes, 20 minutes, there will be no starch, so that's why we will not give the blue-black color. In this case, the enzyme has got destroyed. So after 5 minutes, after 10 minutes, you check it after 10 minutes, add iodine solution, no blue-black color. After 20 minutes, put iodine solution, no blue-black color. या देगा नहीं ये तो देगा. आफ्टर 5 मिनट्स हम आयोडीन सॉल्यूशन डालेंगे, ब्लू कलर देगा, ब्लू ब्लैक कलर देगा. ओके, आफ्टर 10 मिनट्स आयोडीन सॉल्यूशन ब्लू ब्लैक कलर, आफ्टर 20 मिनट्स आयोडीन सॉल्यूशन ब्लू ब्लैक कलर, आफ्टर 30 मिनट्स आयोडीन सॉल्यूशन ब्लू ब्लैक कलर. It means here in this case at high temperature, very high temperature, the active site of the enzyme has been destroyed and enzyme has got denatured, and that's why there is no breakdown of starch into glucose at all. So starch is lying there as such, and that's why it gives blue black color after 5 minutes, 10 minutes, 15 minutes, 20 minutes, 30 minutes. So this is the pattern. Now let me come to the demonstration. एक पार्ट हो गया, ठीक है. अब हम दूसरा करते हैं. ऑन कर दियो.
वो वाला हटल नहीं ना
ऑन है. चलो start. Now students, let me demonstrate the practical. effects of different temperature on the activity of salivary amylase. So now we have taken the three you can say types of water bath we have prepared. This is the water bath at very low temperature around 4 degrees Celsius. So ice has been kept into this water, so the temperature is very low, around 4 degrees Celsius. In this case, the temperature is 37 degrees Celsius. You can see the thermometer. The thermometer shows 37 degrees Celsius temperature. The temperature in this is 37 degrees Celsius. In this, the temperature is 37 degrees Celsius. Okay, you see the thermometer. And in this case, the temperature is more, that is, it is more than 70 degrees Celsius. You can see the temperature, it is more than 70 degrees Celsius. Maybe 76 or like that. So what happens at this very low temperature? At this very low temperature, the enzyme activity is very slow at very low temperature. So what happens over here? So we have put this is the test tube containing the starch solution plus enzyme Tyline is there. So you can see the color. You can see the color. You can see the color of this test tube. So what the color? It is a very light blue color you can find in this area. So means digestion of breakdown of starch into glucose has started. So after 10 minutes we are taking the reading. After 10 minutes you can see very light or very small, very less water breakdown of glucose into starch into glucose. So light or faint blue color you can see over here. Now this is the second case, the body temperature. In this case, the body temperature, what happens? So enzyme is active. Say enzyme has, after 10 minutes, the enzyme has broken the starch into glucose. So starch has been broken into glucose and we have put the iodine solution into it. So what the color? The color is yellowish. So iodine color is also yellowish in color. So there is no blue color in this case. It means starch is not present. Starch has been broken down into glucose by the enzyme salivary amylase at body temperature 37 degrees Celsius. Okay. Now this is the third case. In this case the temperature is very high. The temperature is more than 75 degrees Celsius temperature. So what happens? So in the very beginning, after 10 minutes, the enzyme became denatured. Enzyme got denatured in this test tube. Starch solution plus enzyme. Okay. So enzyme got denatured. So in the absence of enzyme, damaged enzyme, okay, starch is not converted into glucose. Starch is not broken into glucose, so starch is present. So that's why after putting the iodine solution, you can get the clear blue black color. You can see. So enzyme has got denatured. So starch could not be broken into glucose. So starch gives blue black color. What happens in this case? In this case, starch is what? Not there. So in the presence of enzyme, the complete starch has been broken down into glucose, and glucose does not give blue black color after putting the iodine solution. You can see the iodine color. Okay, and in this case the activity of enzyme is very slow, so you can find a little bit of iodine, a little bit of change of color. So a little bit of bluish color you can find in this. So enzyme is working there, but the activity of the enzyme is very slow. So this way we can say that at different temperature the activity of the enzyme is different. At very low temperature the activity of enzyme is very slow. At body temperature, normal temperature, that is 37 degrees Celsius, the activity of enzyme is full and the starch has been broken into glucose completely, and that's why this does not give blue black color. You will get the iodine color only. So color of iodine will not change. In this case, there is no activity of enzyme. At very high temperature, enzyme gets denatured, so the enzyme activity stops, and that's why enzyme could not break starch into glucose, and that's why you are getting the clear blue black color due to presence of starch as such. So this is the experiment. We have discussed the activities of what enzyme at different temperature, around 4 degrees Celsius, around 37 degrees Celsius, and around 70 to 80 degrees Celsius. Enzymes get denatured. Okay, that's all for today. So you note down the practical. I will send you the sample practical. You have to copy it as such in your record book. Thank you. That's all.